Now we're ready to continue with Part C. We'll look at the synapse. If you have not done the first part of the neuron, entitled The Nerve Impulse, be sure and complete that video first. The synapse, as you will see, is the key to the operation of the nervous system. It's more complex than this simplified diagram. A synapse may involve the axon of one neuron and the cell body of another. It may involve the axon of one and the dendrite of another. It may involve two dendrites or two axons. Let's look at the structure in more detail. Here we have a chain of communicating neurons, a pathway. Let's magnify the junction between two neurons. In this view, you have a presynaptic neuron, that's the sending cell in cell-to-cell communication. It contains vesicles, tiny sacs. The sacs contain transmission molecules, neurotransmitters. It contains calcium channels. Then there's the post-synaptic neuron. It's a generator region of the neuron. It contains receptors. The synaptic gap separates the pre- and postsynaptic neuron. The diagram is a little misleading. It's not wide as shown. It's actually not much wider than the width of a single cell membrane. One-to-one presynaptic to postsynaptic neurons generally does not exist. Usually there are many pre to one postsynaptic neuron. Imagine how many synapses the neuron below would have. If you count the tips of the dendrites, there may be 50 or 60 synapses. Let's look at the general function of a synapse. A nerve impulse traveling down the presynaptic neuron reaches the tip of the axon. It stimulates the calcium channels to transport calcium into the axon, and the calcium acts then on the transmitter, on the vesicle, causing release of the transmitter. The transmitter diffuses across the tiny gap and binds to a ligand-gated channel on the postsynaptic neuron. The binding acts as a stimulus on the postsynaptic cell. Does the postsynaptic neuron automatically generate a nerve impulse? No. The graded potential results. It's called a postsynaptic potential, PSP. The transmitter must be removed. After all, the synapse has to be turned off as well as turned on. It's removed in various ways. Degraded by an enzyme. Absorbed by the presynaptic neuron. This is called reuptake. Absorbed by the postsynaptic neuron. The mechanism in the postsynaptic neuron may involve an ionotropic effect. Ion channels open. The receptor is an ion channel. It tends to be fast, found, for example, in neuromuscular junctions. The other type is a metabotropic effect. The receptor is an enzyme that produces a second messenger that may open ion channels and amplify the response. This is slower, as in the secretion of glucose by a target cell. The graded potential in the postsynaptic neuron may be an excitatory postsynaptic potential, EPSP. A depolarization occurs. Graded potential moves closer to threshold. It increases the odds of generating an action potential. It may be an inhibitory postsynaptic potential, IPSP. Hyperpolarization occurs. The graded potential moves further from the threshold potential. It decreases the odds of generating an action potential. Still looking at the general function, summation occurs in synapses. Summation means to add postsynaptic potentials. Temporal summation is one type, 2 or more stimuli acting on one presynaptic neuron. The stimuli occur at different times, full milliseconds apart. Temporal refers to time. Before the graded potential can drop back to resting potential, the second stimulus acts on the neuron, opening more sodium channels, adding a little bigger graded potential. The spatial summation, on the other hand, it involves 2 or more synaptic neurons that act on the neuron. Spatial means space. The synapses are separated by space. Here's a simple illustration involving spatial summation. Notice you have 3 presynaptic neurons synapsing with one postsynaptic neuron. Of the 3, let's say number 1 is excitatory, number 2 is excitatory, but number 3 is inhibitory. Let's assume further that the magnitude, that is the height, of the resulting graded potential is the same for each. The graph shows possible graded potentials in the postsynaptic neuron. For reference, number 1 is excitatory, number 2 is excitatory, number 3 is inhibitory. Which graded potential in the postsynaptic neuron would result if a nerve impulse arrives from these combinations of presynaptic neurons? Number 1 only. The graded potential would be illustrated by A. Number 2 only. Again, A. How about number 3? This would result in the hyperpolarizing graded potential, B. What about number 1 and 2? Well, 1 adds to 2, and C would represent the graded potential. What about 1 and 3? Well, 1 is excitatory, 3 is inhibitory. They cancel each other out, so D represents the graded potential. What about numbers 2 and 3 only? That again would be D. What about all 3 of them? If an impulse arrives from all 3, A would result, because an excitatory would be canceled out by an inhibitory, leaving one excitatory. Which graded potential would generate a nerve impulse in the postsynaptic neuron? The dashed line is the threshold potential. That would be C. So you see, it takes a certain combination of incoming nerve impulses to generate a nerve impulse in the postsynaptic neuron. What about drug actions? The synapse, turns out, is a major target of drugs. There are 2 general actions of drugs. Drugs may act as an antagonist, which decreases synaptic activity. That is, it counteracts the action of the transmitter. Or the drug may act as an agonist. It increases synaptic activity and enhances the action of the transmitter. Does an antagonist always decrease the overall activity of the nervous system? Does an agonist always increase the overall activity of the nervous system? No, it depends on whether the synapses involved are excitatory or inhibitory. An antagonist acting on an excitatory synapse will decrease synaptic activity. An agonist acting on an inhibitory synapse will increase synaptic nervous system activity because you're removing one of the brakes. An agonist acting on an excitatory synapse will increase nervous system activity. But if the agonist is acting on an inhibitory synapse, the general result is a decrease in nervous system activity. Synaptic activity may be decreased by blocking the entrance of the calcium channel, preventing transmitter release, blocking the receptors, increasing the removal of the transmitter. Synaptic activity may be increased by preventing removal of the transmitter, mimicking the transmitter by binding to the receptors. The effect of drugs on the impulse frequency: if it's excitatory, the impulse frequency increases. If it's inhibitory, the impulse frequency decreases. Let's look at the classes of synapses. Best understood are the cholinergic synapses that use acetylcholine as a neurotransmitter, and the adrenergic synapses. They use mainly norepinephrine. Others include histamine synapses that use histamine, amino acid synapses that use various types of amino acids. Peptide synapses use several peptide neurotransmitters, and there are many, many others. Let's look at some details, beginning with cholinergic synapses. These are named for the choline part of acetylcholine. The neurotransmitter is, of course, acetylcholine. It's produced in the cell body from acetyl-CoA plus choline. It uses an enzyme called choline acetyltransferase. It's transported from the cell body to the tip of the axon by microtubules in anterograde transport. It's generally excitatory throughout the nervous system. It's found in the central nervous system, the autonomic nervous system, and of course in the neuromuscular junctions of skeletal muscles. It's removed by an enzyme called cholinesterase, or ACHASE. It's hydrolyzed to acetate and choline. The choline is taken back up by the presynaptic neuron, transported to the cell body, and used to manufacture more of the neurotransmitter. There are two major types of cholinergic synapses. One type are the muscarinic synapses. They're found in smooth and cardiac muscle. They delay the opening of potassium channels. And notice what happens. When that happens, if the channels don't open quick enough, the graded potential remains above threshold longer. So it extends the graded potential and causes a prolonged contraction of the muscle. The other major type are the nicotinic cholinergic synapses. They're located in skeletal muscle cells, autonomic preganglionic neurons, and the adrenal medulla. So you'll understand more about these as we progress. What these channels do is open the sodium channels faster, and so the graded potential reaches threshold quicker. It causes a fast contraction of skeletal muscle. Let's look at some drug actions. Muscarine is a mushroom poison that mimics acetylcholine at muscarinic synapses. It may also be used as a nerve gas antidote. Atropine is a competitive antagonist of muscarinic synapses. Botulism toxin, a bacterial toxin, it decreases acetylcholine release at neuromuscular junctions. Specifically, these are the nicotinic synapses. Skeletal muscle contraction, therefore, decreases. Major symptom would be respiratory failure due to paralysis of the breathing muscles. Nicotine mimics acetylcholine at the same synapses, thus increasing sympathetic nervous system activity. And as you'll see, this is involved in coping with stress. Curare comes from several South American plants, is a competitive antagonist of nicotinic acetylcholine receptors. Major symptom is respiratory failure, again, due to paralysis of the breathing muscles. Many nerve gases, for example, sarin and other neurotoxins, increase the cholinesterase breakdown to decrease cholinesterase concentrations. This then increases the action of acetylcholine, symptoms of which will be described when we get to the autonomic nervous system. What about the adrenergic synapses? Well, these are named from the alternate term for epinephrine, which is adrenaline. The neurotransmitters are catecholamines. It's a type of monoamine. They include epinephrine, norepinephrine, and dopamine. Other monoamines are serotonin and histamine. Notice the similarity of tyrosine below with norepinephrine. Norepinephrine, we'll abbreviate it NE, is produced from tyrosine, which is transformed into dopa and then into dopamine, and from there transformed into norepinephrine. It's both excitatory and inhibitory, depending on the synapse. Its location, sympathetic nervous system, the brain stem, a part called the reticular formation, and the hypothalamus, the cerebellum, and the cerebrum. Smooth muscle, cardiac muscle. It's removed by reuptake, stored in vesicles, and then broken down by monoamine oxidase in the terminals of the axon. It's broken down also by catechol-methyltransferase, so there are several enzymes that help break it down. The major types of these synapses are alpha and beta. In general, the alpha and beta 1 are excitatory and the alpha and beta 2 are inhibitory. Alpha synapses contract smooth muscle. Beta 1 contract cardiac muscle with greater force and spread. Beta 2 relax smooth muscle. A drug called monoamine oxidase inhibitor increases the force of contraction of the heart. Why would that be? Because the monoamine oxidase breaks down norepinephrine. If it's inhibited, the norepinephrine remains in the gap longer and exerts its effect longer. Epinephrine. It's an excitatory and inhibitory neurotransmitter. Its location is mainly in the brain stem. Also a hormone secreted by the adrenal medulla. This is commonly called adrenaline. Dopamine is yet another. It's excitatory and inhibitory. It also gives a sense of pleasure. It's located in the basal ganglia, the limbic system, the brain stem, and something called the hypothalamus pituitary system. We'll discuss these a little later. Again, look at drug actions. Cocaine blocks the reuptake of dopamine to enhance the sense of pleasure. Without reuptake, no recycling to make more dopamine occurs. The supply of dopamine therefore decreases and the pleasure is replaced by depression and anxiety. Many antidepressants, such as Prozac, block serotonin reuptake. Some tricyclic antidepressants block norepinephrine reuptake. It's not as commonly used as serotonin reuptake inhibitors. Amino acids make up another broad class of synapses. A common one is called GABA, gamma-aminobutyric acid. It's an inhibitory synapse, that is, it causes a hyperpolarizing graded potential. It's located mainly in the basal ganglia, but widespread in the central nervous system. The receptor is a chloride ion channel. It increases the transport of chloride ions into the cell, causing an IPSP, inhibitory post-synaptic potential. It decreases the chances of generating a nerve impulse. It results, therefore, in a decrease in overall nervous system activity. Drug actions may act on GABA receptors. The receptor contains sites for alcohol, Xanax, and other substances. Therefore, substances like Xanax act as a tranquilizer, and alcohol depresses nervous system activity because they both bind to the receptor. Other amino acid synapses include those for glycine. It's generally an inhibitory neurotransmitter. Glutamic acid or glutamate, very common in the brain and central nervous system. It tends to be excitatory. It's important in vision. Drug actions related to these include tetanus toxin. It blocks the release of GABA and glycine. It's transported by the lymph and blood to neuromuscular junctions, absorbed into the neurons, and transported to the central nervous system by retrograde axonal transport. Lack of GABA and glycine allows an increase in general muscle contractions. Another group of synapses are those involving peptides. These are also called neuropeptides, brain peptides, or opioids. There's one called the beta endorphin. This is a group. General function is pain relief. It's located in the limbic system, the spinal cord, the thalamus, the hypothalamus, the brain stem, and the GI tract, the gut. Enkephalins, another one, also function as pain relief, located like the endorphins in the same groups of organs. Serotonin, this is another monoamine. Its general function is also pain relief. Other neuropeptides include many hormones, which we'll cover in later units, such as insulin. When people started understanding more about drug actions on the brain, they wondered why in the world does the brain have receptors for opium and other such substances? Well, let's look at a few of those. Commercial forms include hydrocodone, oxycodone, codeine. They function to relieve pain. They may depress the respiratory system. Drug actions on these synapses include narcotics. They increase the activity of peptide synapses. Naloxone is a narcotic antagonist that blocks opioid receptors. It's used to treat narcotic overdose and reverse respiratory depression caused by narcotics. Generally, it's injected. Some is administered by a nasal spray, such as Narcan. Well, after they began to wonder about all of this, it was discovered that in the brain and the central nervous system are naturally occurring opioids, the peptides we're talking about, and it just happens that the drugs that we discussed act on the normal receptors. What would treatment with naloxone do to the sensation of pain, and why? Well, it would tend to increase pain sensation because it inhibits the action of the opioids. Other neurotransmitters include ATP, nitric oxide. This is unusual because it's a gas. It's located in the central nervous system, and it sends neurons into the penis. It's produced using an enzyme called nitric oxide synthase. A major role is to relax smooth muscle. Carbon monoxide is another neurotransmitter. It tends to be excitatory, and it's produced in the brain. We have now completed Unit 11, Cells of the Nervous System. You're ready to progress to Unit 12, the Functional Nervous System. Begin with Part A.